Hi, I just wanted to say thank you for having me and allowing me to give this talk. So my name is Kori Kuzma. I'm a software developer at Nationwide Children's Hospital in Dr. Alex Wagner's lab. And yeah, I'll be talking about how we can translate human readable variation descriptions to unique computable variations with the Variation Normalizer. So this is a project under the Variant Interpretation for Cancer Consortium, also known as VIC. And VIC is the driver project of the Global Alliance for Genomics and Health, also known as G4GH. And the mission is to standardize the creation, representation, and interpretation of clinically relevant evidence associated with genomic variation in cancers. So this figure demonstrates variant evaluation and interpretation. In step 1, variation is assayed in a lab, and we get assay-specific outputs that are stored in a preliminary variation report. In step 2 to 3, we highlight how the clinician, curator, or variant scientist must perform a significant amount of manual effort. In step 2, they annotate the preliminary variation report with HTTPS, ISCN, or other nomenclatures. In step 3, they must map the preliminary variation report to heterogeneous datasets or knowledge bases. And finally, in step 4, they make their final evaluation as a clinical report and study. And we can see that the human evaluation of variation can be labor-intensive, inconsistent, and prone to error, which could create a bottleneck. Here in the top row, we can see a list of different biomarkers, such as genotyping, gene fusions, and RNA expression. Each of these biomarkers can be assayed by a number of different assay technologies, such as histochemistry, PCR, and sequencing. And each of these assay technologies can use different variation calling methodologies, such as expression estimation, sequence junction detection, and SNV-Dellens calling. And as a result, the observed variation can be represented in many different formats. There is also a related problem of how variation is described in knowledge bases. Here we can see an example of a variant in the civic knowledge base. It can be represented as aliases, HTTPS expressions, and centralized authoritative IDs. And it can be challenging to determine the relationship. Are all these representations talking about the same variant? Now that I've introduced you to the problem, I'm going to introduce you to verse and versatile frameworks, which can help solve this problem. The Variation Representation Specification, or VERS, is an open specification to standardize exchange of variation data. It provides models for representing systemic and molecular variation, an algorithm for generating globally unique computed identifiers, and fully justified allele normalization algorithm. And all of these allow for a way to represent semantically precise and unambiguous representations of variation. Here we can see a very similar diagram as before. However, now this includes verse. Step 1 is the same. However, the labor-intensive steps are now replaced by verse. So in step 2, we translate to machine semantics using verse computable representations. And in step 3, we can use these verse computable representations and help us do the mappings to the heterogeneous knowledge bases and data sets. And now in step 4, we can computationally store these results in a database. And in step 5, we can have the human readable reports exported in a clinical report and study. And in step 6, we can highlight how the clinician, curator, or variant scientist now only has to review and validate. They no longer have to do a lot of labor-intensive steps. So we can see that using computable standards can help improve the efficiency of variant assessment and interpretation. Verse added tools for interoperable loquacious exchange, or versatile, is a set of proposed extensions for verse to enable practical, real-world data exchange of variation. Here's an example how we can decorate verse variation using versatile. In the middle, we can see a verse allele object, which is represented by a computed identifier. We can annotate this verse allele using versatile and provide additional information, such as alternate labels, expressions, or external identifiers. And we can see that versatile enables resources to richly describe verse variation. Now that I've introduced you to verse and versatile, I will be introducing the Variation Normalizer and how we can represent variations using these frameworks. So the Variation Normalizer normalizes and translates human readable variation descriptions to verse and versatile representations. You can see our API on the right, which is found at normalize.cancervariants.org/variation. You can see that there are so many different endpoints, so today I'm just going to be focusing on the normalized endpoint. So what exactly does the normalize endpoint do? Well, you can see the list of related variant contexts with multiple representations, such as free text queries using 1-letter or 3-letter amino acid codes, HDVS expressions used on different accessions and positions. And choosing one as a representative is a challenge that clinical labs face. The normalize endpoint provides a mechanism from community standards such as MAIN in order to provide a consistent computable identifier, which is highlighted in the red box. Now that I've introduced you to what the variation normalizer is, I'm going to dive into the fun part on how exactly it works. The first step is tokenization, which splits the input string into smaller units, also known as tokens. First, we take the initial query, braf v600e, and split onto white space to give us two separate strings, braf and v600e. To the machine, this is just a string of characters. There is no meaning. It's just 4 characters in a string. This is where tokenize can help us. We can use tokenize to create computable entities to give us some meaning to these strings. So we can see that braf represents a gene token, and v600e represents a protein substitution token. From tokenization, we return an ordered list of tokens, a gene token and a protein substitution token. We then want to classify these tokens. This means that we're trying to know what kind of variations these tokens represent. And each of our classifiers has a list of ordered tokens required to be classified as that variation type. We can see that our ordered list of tokens do not get classified as a protein deletion, since we do not have a protein deletion token. However, it does get classified as a protein substitution token since we have both a gene token and protein substitution token. We then move on to validation to ensure that the initial query is valid. We use UTA tools, which pulls from REST-seq and Ensemble data, in order to get all the protein accessions associated with the gene, BRAF. We then perform some simple validation checks using Biocommon seq repo, which gives us fast access to sequence data. We return a list of valid and invalid results. In the first, we can see the accession. We found V at position 600, which is what we're expecting, so it's valid. In the second accession, we found T at position 600, which is not what we're expecting, so it's invalid. Now we move on to the most important step where we choose our representative transcript. So we have our valid result, and now we're going to map to the genomic annotation layer. And we use Biocommon's UTA database to help us do this. It contains transcript and alignment data. We'll then use PyLiftover to lift over to the GRSH38 assembly. And now we move on to selecting our preferred compatible annotation. This is where we make some decisions on our transcript priority. First, we try to find main select, then main plus clinical, and then the longest compatible remaining transcript. And you can see in this example, we found a main select transcript, and now we're going to map back down to the starting annotation layer, which is the protein level. Now we move on to translating to the versatile representation. We're going to perform fully justified allele normalization using verse Python and translate to versatile. Again, we're output with the verse allele, which can be represented by a computed identifier. And we can annotate using versatile and provide additional information, such as the molecule context, the structural type, and the verse ref allele sequence, as well as the gene context that we get from our gene normalizer. Now that I've introduced you to what the variation normalizer is and how it works, I'm now going to be talking about the application that it's used in. Variation normalizer is used in MetaKB, and in MetaKB v1, it was found that 6 somatic cancer variant knowledge bases are highly disparate in content, structure, and supporting primary literature. And this could create a bottleneck when evaluating variants and their relevance in a clinical setting. So Framework was developed for harmonizing interpretations to produce a knowledge base of over 12,000 aggregate interpretations. We've continued to iterate on MetaKB post v1. We now harvest evidence data from Civic and Moamnak, and we transform this harvested data to a common data model with the help of our various normalizer tools, which are open source. And this includes the variation normalizer. We then store this transformed data into a Neo4j GraphQL database. Here's an example of a query that you can make in MetaKB, which relies on the variation normalizer. In this query, we're trying to find all the statements or evidence items where the variation is BRAF v600e. Here are 2 of the many evidence items that are returned in this query. On the left is a Civic evidence item, and on the right is a Moamnak assertion item. Each of these knowledge bases represent BRAT v600E differently, but the variation normalizer is able to return the same computed identifier. So we can emphasize that the verse computed identifier allows us to do easy lookups of variation, even when represented differently across knowledge bases. So our future plans would be to add support for other variant types, such as categorical variation. We also want to provide translations from other nomenclatures, because we currently only support HGVS and Nomad VCF. We also want to improve the performance on free text queries, because it takes a bit longer than our HGVS expressions. I'd like to thank my collaborators for all their contributions to this project, NIH for funding, and again, thank you CGC for having me and for the travel award. You can scan this QR code to find our API, our open source GitHub repo, more information on the verse and versal frameworks, as well as my contact information and a PDF of the slides. Thank you.